Okay, so I'm going to do some of the introductory type slides from chapter 27, which is the antimicrobial chapter. So looking at chemotherapies, drugs that are used to treat bacterial, viral, and fungal infections. And again, I'm just going to try to give you some of the introduction type information so that you can probably have a better understanding of some of the terms that are used on that antimicrobial worksheet that we will be completing the activity with on Wednesday's tomorrow's class. So with antimicrobials, what we're looking at are drugs that are used to treat bacterial, viral, or fungal infections. Antimicrobials is a catch-all term that includes antibiotics, antivirals, and antifungals. So the more specific terms would be an antibiotic, which is used to treat a bacterial infection, an antiviral that's used to treat a viral infection, and an antifungal that's used to treat a fungal infection. So if you take an antibiotic, it's not going to treat a virus. So if you were sick with the flu and you took an antibiotic and you feel better, it's because you were actually feeling better from the flu. So they work against a specific type of microorganism. If we look at antibiotics, they're the first antimicrobials that were really identified. And generally what happens is that they were identified from a natural source. So we want to keep in mind that microbes are competing with each other. And so one microbe might produce a chemical or a compound that will actually inhibit the growth or kill another microbe. And so what happened early on was Dr. Fleming here, which is pictured, he was known actually as an artist, not only as a doctor, but he used microbes to paint with. And so, as you know in lab, colony morphology, one of the characteristics of colony morphology is pigments. And so he was actually very talented in using different microbes that produce different pigments, and he was able to time their growth so that the pigments would be, at the same level that he wanted for his pictures. And so this was a very difficult skill to master. So he was actually the president of an art group in Chicago. And he's one of those that kind of borderline, is he a scientist or is he an artist? Well, in culturing his different bacteria that he had, what he noticed was that there was this area of inhibition of growth of his Staphylococcus aureus. And so the story goes was, he was eating a sandwich and some of the mold from the sandwich fell off onto the plate or whatnot. So there's a version where there's a sandwich involved, and there's a version where he was just culturing it on an agar plate. But he noted this inhibition of growth, and what he was able to do is then characterize the fungus that was growing was penicillium. And further work was done where the compound penicillin was actually isolated from the penicillium, the microorganism. So the work that was done by Fleming was back in 1928, and this really obviously had a huge impact on how we could then treat bacterial infections. The further isolation purification was done in 1940. In the 1930s, in between Fleming and the purification by Fleury and Chain, sulfa drugs were discovered. So this is another antibiotic that's used for bacterial infections. With sulfa drugs, it inhibits vitamin synthesis, so a different mechanism than penicillin. And then further in the 40s, streptomycin was identified. These were all identified from natural sources, so microorganisms, either soil bacteria like we're isolating in the lab, so either soil bacteria or molds, funguses, again, identifying chemicals that are present in them, then that we can isolate and then use. From these natural antimicrobials, what has been done in the lab is that the bonds the side chains present on these compounds have been changed and altered, and that's what's given us these different generations of drugs. So, penicillin was the first in the penicillin family, but then if we alter the side chain, we then can get amoxicillin, ampicillin, methicillin, the other members of the penicillin class or family. So we want to remember with an antimicrobial or an antibiotic that it has to have selective toxicity. So yes, it needs to be toxic because it needs to kill the microorganism, but we want to make sure it has minimal to no effect on humans. So some of these might have side effects in high concentration, but if we're giving it a pharmacological dosage, maybe a lower concentration, that it should have minimal side effects to humans. One of the terms that's on your antimicrobial worksheet is spectrum of activity. And spectrum of activity is a term that's more widely used with antibiotics, so with bacterial infections. So if you do have a one that's antiviral, antifungal, this term doesn't work so well. You might want to look, does it work against a specific virus or does it work against all virus? The idea with narrow spectrum is that it has a very specific organism or a very limited number of organisms that it would work against. So for bacteria, a narrow spectrum would be like penicillin because it works well against gram-positives, and that's because of the mechanism of action, how it works. So gram-positives have that thick cell wall, and so there's more targets for the penicillin to work against. But gram-negative has a really thin cell wall, and so it's not going to work very well against gram-negatives. Okay, and so again, penicillin, a good example of a narrow spectrum antibiotic because it works well against one group, one type of bacteria, but not all of them. Now, a little caveat, if you Google penicillin or amoxicillin, you'll see references that it does work against gram-negatives. So sometimes when in the lab they alter the drugs to get the different generations, the initial spectrum of activity changes over time as they've changed those side groups. The cephalosporins are a good example of that. So cephalosporin, the first generation, works really well against gram-positives, but as you go down to your 5th, 6th generation, those don't work against gram-positive anymore, but work against gram-negatives. And then we don't know why. It just does, and sometimes in medicine we just use what works and we don't question the mechanism. Now, again, narrow spectrum, small group. Broad spectrum is where it would work against a number of different organisms, kind of like a catch-all. So examples of broad spectrum would be like tetracycline, doxycycline. They work against gram-negatives and gram-positives. So a lot of times when we have emerging diseases we're not sure what's causing it, you'll use a broad spectrum because regardless of what it is, it will treat it. And so when Lyme disease first came about, they didn't know what was causing Lyme disease. They honestly didn't know whether it was bacterial or viral, and they said, well, give them tetracycline and see what happens. Well, for at least a good majority of the patients, tetracycline was treated the disease very well, helped them manage signs and symptoms of the disease, and so it was kind of told us to go down the rabbit hole of bacteria, not virus, and further identification of the microorganism led to that it was gram-positive. Now, tetracycline is still used for Lyme's disease, and that's just kind of, you have to retrain the physicians, and that takes time. In some cases where tetracyclines can't be prescribed, such as young children, penicillins, like amoxicillins and such, will be prescribed because, again, it's a gram-positive microorganism. So again, you should look at your antimicrobial that you've been assigned and try to determine, is it narrow spectrum? Does it only work against a handful of microorganisms or specific groups of microorganisms, being like specific bacteria? Broad spectrum, it works against like all bacteria or all viruses, all fungus. Another term that is used for antimicrobials and antibiotics, more specifically, is this idea of whether the drug works as a static or Seidel. So static means that the microorganism's not able to reproduce. It doesn't mean that the microorganism has been killed. It just can't grow and divide. So it's kind of static. It's frozen. It's not actually like freeze or frozen, but it can't grow and divide. It can't replicate. With Seidel, it means that the microorganism is actually killed. So the cell gets lysed, damaged, it's destroyed. And so these two different modes, the static versus Seidel, both can achieve microbial death. Okay, and the term microbial death kind of is an oxymoron because it doesn't always mean death, right? So it includes static, and again, if you have something that's bacterial static, it just causes the bacteria not to be able to reproduce. A lot of times this is because it halts protein synthesis or DNA synthesis. So the cell's still alive, but it can't make more protein. With something that's bacterial static, if you treat for a long enough time, sometimes you can induce Seidel. So if a bacteria goes 2 weeks without making a protein, eventually it's going to die. So your tetracyclines are a good example of something that's bacterial static because they block protein synthesis. So they don't kill the cell right away, but if you take a long enough course, and the normal course of treatment with something like tetracycline is 14 days, in some cases even longer, 21 days, and in some cases with chronic Lyme's, they're even prescribing 4 months. Obviously this has other implications as far as side effects though. So going back to your static and your Seidel, there's a prefix that goes to the static and Seidel. So if you're talking about an antibiotic, it would be bacterial static, bacterial Seidel. If you're talking about an antifungal, fungal static, fungal Seidel. If it's a virus, viral static, viral Seidel. And the more general term would be microstatic, microcidal. Okay, so those are all different terms. You know, for exam, if I were to say, if something is fungicidal, what does that mean? You should be able to kind of define what that term means. How does that relate? Like, what would be the target organism? Would it be killed or not being able to reproduce? Okay, and then, so lastly, what I want to do is kind of introduce the different mechanisms of action. And so when we look at mechanism of action, this is how does the antimicrobial work. And these mechanisms of action are more specific to antibiotics. So just keep that in mind. Sorry, antiviral, antifungal people. When we have a good antibiotic, it needs to target something that's different. And different between them being the bacteria and us being the host. So if we target something that we have that's shared, there's going to be more side effects. If we target something that's different, it's going to affect their fill in the blank and not our fill in the blank. And so if we think about the different cell structures that are present, one of the unique cell structures that we both have, but they're different, are ribosomes. So the ribosomes on bacteria are 70S. Our ribosomes are 80S. So there's a size difference in the ribosomes because the subunits that make them up are different. And so if we have a drug that affects the 70S ribosome, it's going to have low to no side effects to us. Now, the caveat of that is our mitochondria actually have 70S ribosomes, so some of the side effects we see is if the drug gets all the way into the mitochondria. Now, that's where if you take a high dose, if you have a high concentration, it's more likely to actually migrate into the mitochondria and have an effect. So we see protein synthesis is one of the mechanisms of actions that's pretty common. The other thing is we humans, we don't have cell walls. Bacteria do. and so we can target the cell walls, and so that's another area of mechanism of action that we see widely used with antimicrobials. So I'm going to move on to the table. So this is a table from your textbook, and as you can see, cell wall synthesis, we have a number of different antibiotics that work on blocking cell wall synthesis. If a microorganism, if a bacteria can't make a cell wall, then the cell wall provides structure to them and allows them to maintain their shape. And so if we damage this, if it's not able to repair, if it's not able to make new ones, new cell walls to support growth, the cell dies. Okay, you can see protein synthesis is listed next as a target, and so again, this targets that ribosome, the difference in the ribosomes. So things like the tetracycline, it's going to bind to the 70S ribosome and not to the 80S. Aminoglycosides too. So we can also have a mechanism of action of cell membrane. So polymyxin works off of that. Also, our antifungals tend to be cell membrane targets. We do have to keep in mind that we have cell membranes, and so with antimicrobials that fall under a mechanism of affecting the cell membrane, there tends to be a higher toxicity because we have cell membranes. There are also another mechanism of action is affecting nucleic acid synthesis, DNA synthesis, and RNA synthesis. So kind of large category here. Our antivirals in some cases fall under this category because think about a virus has to replicate its genetic information, and it's using the host machinery. And so if we affect the host cell's ability, that's being our cell's ability to actually make nucleic acids, then that's going to affect the virus being able to replicate and then increase in the numbers and then being spread to other cells. Now, again, this is a good antimicrobial affects something that's different. Well, the virus is using our machinery, and so it's going to affect our machinery. And so with a lot of these antivirals, we see a lot more side effects, and so sometimes it's better just to let the virus clear versus take the antivirals. Okay, now nucleic acid inhibition can also be a mechanism of action for antibiotics too. Intermediate metabolism, sometimes this is called vitamin synthesis in other textbooks, and so our sulfas fall under this category. And so sulfa drugs actually affect the ability to produce folic acid, and folic acid is important for DNA synthesis and also protein synthesis. And so this is why women who are pregnant are given, are told to take vitamin supplements with folic acid, because the fetus is growing all these cells, increasing numbers and proteins, and so the demand for folic acid for pregnant women is obviously much higher than anyone who's not pregnant. So we don't make our own folic acid. We lack the enzymes required in order to take PABA, which is the substrate for the reaction, and to make folic acid. So we as humans require to either get that through our diet or through supplements. Well, bacteria have the enzymes in order to make folic acid. They don't have the luxury of always depending upon a meal coming in with folic acid. And so they have those enzyme systems, so we can target those enzyme systems, and that's what the sulfas do. And again, as I mentioned before, sulfa's been around since 1930, so they are kind of a drug that's used quite commonly.